Welcome to the deep dive. Today we're getting into something really fundamental to how we age. It's called proteostasis.
Mm-hmm.
Think of it like this. Your cells are these complex orchestras, right?
Okay.
And the proteins, they're the musicians. Each one has to fold into a specific shape to play its part.
That's a great analogy.
But, what if some musicians start playing badly? Or can't even put their instruments together, right?
Right, that's the problem.
That's where proteostasis comes in. It's sort of the conductor, making sure everything's harmonious, and maybe the repair crew too.
Exactly. It's the whole quality control system for proteins.
So today we're going to explore how our cells keep this protein balance, what messes it up as we get older, and some really exciting new ideas about how we might actually be able to tweak this whole system.
Yeah, the orchestra analogy really works. And the research we looked at, it really drives home how crucial maintaining this protein harmony is for healthy aging.
It sounds incredibly complex.
Oh, it is.
Yeah.
The system cells have evolved are incredibly sophisticated.
Yeah.
Making sure proteins fold right, getting rid of the ones that don't. It's a constant job. And these systems can weaken over time.
Okay, let's try to unpack this. Where does it start? The sources mentioned the ER proteostasis network. That's the endoplasmic reticulum.
That's right, the ER. It's where a huge chunk of our proteins, maybe about a third, actually get folded into their final functional shapes.
So it's like the main rehearsal room for these protein musicians.
Exactly. But it's not passive. It's got this really dynamic quality control system built in. It's called the unfolded protein response, or UPR, specifically UPR-ER, because it's in the ER.
UPR-ER. Got it.
And the fascinating thing is it has these three main sensor proteins constantly monitoring the situation. They're called IRE1, PERK, and ATF6.
So sentinels in the rehearsal room, what do they do if they spot trouble, like too many unfolded proteins piling up?
Well, when they sense that stress, that buildup of misfolded proteins, they kick off this really coordinated response. It's sort of a three-pronged attack.
Okay, what's the first prong?
First, they dial back the overall production of new proteins, less the load on the system,
Makes sense. Don't make more mess while you're cleaning up.
Exactly. Second, they ramp up the production of what are called molecular chaperones. These are like helper proteins that assist in proper folding.
Folding assistance. Okay.
And third, they activate pathways to clear out the misfolded proteins that are already there, the rogue ones. Get rid of the problem proteins.
For reduce workload, get more help, and clear out the garbage. Seems logical.
It's a very effective damage control system.
Now, here's something I found really interesting in the sources. It's not just about crisis management, is it? It seems like having this UPRA system ticking over at a moderate level is actually good for long-term health.
That's a key insight, especially in tissues that don't turn over very quickly, like the heart and the brain. This sort of ongoing low-level UPR-ER activity seems to help maintain their protein folding capacity.
Like keeping the orchestra match fit all the time.
Yeah, that's a good way to put it. It helps delay that age-related functional decline in these really critical tissues.
And this has led to exploring ways to, support the system therapeutically.
Absolutely. That's where it gets really exciting. There's research into using chemical chaperones. One example is something called sodioxycholic acid, or 2-UDCA.
UDCA, okay.
And even gene therapy approaches are being explored. The idea is to deliver more of these UPR regulators, like those sensors IRE1 or PERK, directly to the cells.
Wow. And has this shown any promise?
Yes, actually. In studies with aged mice, these kinds of interventions have shown they can reverse some age-related defects in certain tissues. It's still early days for human applications, of course, but it's very promising.
Okay, so boosting the ER's maintenance crew is one angle, but the sources also really zeroed in on another player, NRF2. Sounds like a bit of a multitasker.
Oh, definitely. NRF2 is a really central regulator. It defends against two major cellular stresses simultaneously.
Which are?
Oxidative stress that's damaged from reactive oxygen species, ROS.
Right, the free radicals we hear about.
Exactly. And also proteotoxic stress, which is the stress caused by the buildup of those misfolded or damaged proteins we've been talking about.
So it bites on two fronts. How does it actually work?
Well, normally NRF2 is sort of held captive by another protein called Key-Beel. It keeps it inactive.
Yeah.
But when the cell senses stress, either too many ROS or too many misfolded proteins, NRF2 manages to escape from Key-Pool.
Breaks free.
Breaks free, yeah. And then it travels into the cell's nucleus. And once it's there, it acts like a master switch,
Flip it on.
flipping on a whole battery of defensive genes. These genes code for things like enzymes that make our own powerful antioxidants. Glutathione is a key one.
Ah, boosting internal defenses.
Right. And it also activates genes for building blocks of the proteasome.
That's the cell's protein shredder.
Exactly. The machinery that breaks down damaged proteins. And it also switches on factors involved in autophagy.
The cell's recycling system.
Correct. Another crucial way cells clear out damaged goods, including proteins. So NRF2 coordinates antioxidant defense, protein degradation, and recycling.
That's quite a portfolio. But, the sources mentioned something about NRF2 declining with age.
Yes, that seems to be a common theme. NRF2 signaling tends to become less robust as we get older. And the thinking is, this isn't just random wear and tear. It might be linked to things like chronic low-grade inflammation, sometimes called inflammaging, or other age-related shifts in cell signaling.
And the consequence of lower NRF2 activity.
Well, it makes our cells more vulnerable, more susceptible to damage from ROS, and less efficient at clearing out those misfolded proteins. The defenses are weaker.
So researchers are looking at ways to boost NRF2 back up.
Precisely. There's a lot of interest in developing small molecule drugs that can activate NRF2, essentially helping to restore that signaling pathway.
Like giving the system a kickstart.
Yeah, boosting antioxidant capacity, enhancing protein clearance. And these NRF2 activators have shown some real promise in lab models, particularly for diseases where protein misfolding and oxidative stress are big problems, like Alzheimer's disease and certain cardiovascular conditions.
Okay, so we've got the ER's quality control and NRF2's broader defense system. These deal with proteins after they're made, maybe folded wrong. But what about the very beginning? Making the protein itself. The sources mentioned translational fidelity.
Right, this is about accuracy right at the source. Protein synthesis translation is done by ribosomes, the cell's protein factories.
And they sometimes make mistakes.
They do. It's remarkably accurate, actually, but not perfect. The sources mention an error rate around, say, 1 mistake for every 10,000 amino acids added or codons read.
That sounds incredibly low. Does it really matter?
Well, think about how many proteins your cells make constantly over decades. Even tiny error rates mean that mistakes do happen, and the resulting faulty proteins can accumulate.
Ah, especially in cells that stick around for a long time, like muscle cells or neurons.
Exactly. Those long-lived cells don't get replaced often, so they have more time to accumulate these slightly off proteins, adding to the burden of misfolded proteins the cell has to deal with.
And there's a link between how accurate this process is and how long species live.
Yes, that's a really interesting correlation researchers have noticed. Across different species, those with ribosomes that are inherently more accurate, that make fewer mistakes during translation, tend to have longer lifespans.
Wow. So translational accuracy itself might be a factor in longevity.
It strongly suggests it plays a role, yes. And based on this, researchers are actually experimenting with what they call proofreading boosters.
Trying to make the ribosomes even more accurate.
Kind of. Molecules that tweak the ribosome slightly to enhance its accuracy. And in simpler organisms, like worms and flies, giving them these boosters has actually been shown to extend their healthy lifespan.
That's fascinating. Are they trying this in mice yet?
Research is definitely ongoing in mammals, including mice, to see if enhancing translational fidelity can have similar benefits. It's a complex area, though.
Right, because as the sources stressed, these errors aren't the only thing going wrong with proteostasis as we age, are they?
Absolutely not. It's just one piece of a much bigger puzzle. You've still got the declining activity of those chaperone proteins we mentioned.
Folding assistance.
Right. And autophagy, the recycling system, that also tends to become less efficient with age. Plus, the cumulative oxidative damage NRF2 tries to fight. It all adds up.
So multiple things are weakening simultaneously.
Exactly, which logically leads to the next idea that's gaining a lot of traction.
Yeah.
Maybe we need to tackle multiple things at once.
Ah, the proteostasis cocktails idea.
Precisely. Instead of just targeting, say, the UPRR, or just NRF2, or just translational fidelity, maybe combining interventions could be more powerful.
Like a multi-pronged strategy for the whole protein orchestra? What kind of combinations are they looking at?
Well, the sources gave some interesting examples. You could, for instance, combine something that enhances the UPR-ER boosting folding capacity with something that activates the proteasome-enhancing clearance of bad proteins.
Okay, help folding and clean up mistakes.
Or you could combine an NRF2 activator tackling oxidative and proteotoxic stress with something that induces autophagy, ramping up their cycling.
Defense plus cleanup.
Yeah. Another idea is pairing those ribosomal fidelity modulators, reducing errors at the source, with chaperone inducers, helping the newly made proteins fold correctly.
Fewer mistakes plus better folding help. Makes sense. Are these cocktails actually working better in experiments?
The early signs from preclinical studies are quite encouraging, yes. In lab models of things like neurodegenerative diseases or heart problems like cardiomyopathy, these combination approaches often show synergistic effects.
Meaning the combined effect is bigger than just adding the individual effects together.
Exactly. 2 plus 2 equals 5 kind of thing.
Right.
In many cases, these cocktails seem to outperform treatments targeting just a single pathway, at least in these models.
Okay, this all sounds incredibly promising for maybe, extending health span, keeping things working well for longer.
That's the goal, yes.
But it can't be simple. What are the big challenges that still need to be tackled? The sources must have mentioned some hurdles.
Oh, for sure. A major one is delivery. How do you get these treatments, these UPR enhancers or NRF2 activators, to the right place in the body?
Right. You might want to boost proteostasis in the brain, but maybe not everywhere else. Could cause side effects.
Exactly. Precision delivery is key. Getting the therapy specifically to the tissues that need it most, like the brain or the heart, while minimizing exposure elsewhere. That's where things like engineered viral vectors might come in, the viruses modified to target specific cell types.
Okay, targeted delivery. What else? How do we even know if these treatments are working inside the cells?
That's another huge challenge. Biomarkers. We need reliable ways to measure proteostasis health. Can we develop tests, maybe blood tests that can tell us about protein aggregation levels or how active the UPR or NRF2 pathways are in a person?
So we can track if the treatments are actually improving things.
Right. We need good biomarkers to monitor the state of the system and the effectiveness of any intervention. Without them, it's hard to know if you're hitting the target.
And I guess it's probably not a one-size-fits-all solution either.
Definitely not. The sources really emphasize this. The proteostasis needs of, say, your brain are different from your liver, and they change throughout your life.
So an elderly person's brain might need a different kind of support than a middle-aged person's heart.
Exactly. We'll likely need age-specific and tissue-specific strategies.
Mhm.
We need to understand the unique demands on proteostasis in different contexts and tailor the interventions. It's got to be nuanced.
So let's try to wrap this up. It's really clear that proteostasis, this whole business of keeping proteins folded and functional, is just central to aging well.
Absolutely fundamental.
We've touched on the ER's critical role in folding this UPR system.
The first line of quality control.
Then NRF2, that dual action protector against oxidative stress and bad proteins.
A key defensive layer.
And even the importance of just getting it right in the first place, the accuracy of making proteins, translational fidelity.
Right, minimizing errors from the start.
And looking forward, this idea of proteostasis cocktails, combining different approaches, seems like a really exciting direction for potentially extending our health span, keeping that cellular orchestra playing harmoniously for longer.
It really does. By understanding these mechanisms better and finding ways to fine-tune them, often in combination, we're moving towards potentially influencing aging at a very basic cellular level.
This whole deep dive, it really makes you stop and think about the incredible, intricate balance inside every one of our cells. It's just mind-bogglingly complex.
It truly is.
And it makes you wonder, considering this fine-tuned protein balance we've been discussing, what other seemingly small, almost invisible cellular jobs might turn out to have these massive long-term impacts on our health as we age? It just highlights how interconnected everything is, doesn't it?
A very important point. The interconnectedness is key.
Well, thanks for joining us for this deep dive into the world of proteostasis. Lots to think about there.